Alright, in this video we are going to review the central dogma of biology. So the central dogma of biology gets its name because it's one of the most important ideas or principles that you'll ever learn in biology. It basically connects the information that we have in every single one of our cells that we get from our parents, and this happens in animals, plants, and humans. And how does that information become the traits that make us who we are or allow us to live our lives? And so that central dogma of biology is going to connect the DNA into the traits that are our body. And so here are a few key ideas that you got to keep in mind and understand before we go into the steps of the central dogma of biology. So key idea number 1, every cell contains DNA in the nucleus. So if you grab a hair cell or a spit cell or a skin cell, if you go inside of its nucleus, you're going to find an entire set of your DNA. Key idea number 2 is that your body needs proteins to function. And so proteins, what's cool about them is that they have many different shapes. And basically each shape determines what job it does. And for every job that your body needs to get done, there's probably a protein to do it. So for example, in this first protein I'm drawing, maybe this is a protein that helps your body digest sugar. In the second protein, maybe this is the protein that gives your eyes its color. None of these drawings are actually the shape of the proteins, but you're kind of getting the idea. And in this third protein, maybe this is the one that helps your body or helps your skin heal itself if it's been cut. So any job your body needs to get done, it relies on proteins to do it. And so you are alive because of the proteins that are all working really hard. So the central dogma basically connects DNA, which is the starting point of all this, into the end product, which is a protein that helps your body function. And so let's get into the steps of how this happens. How does DNA turn into a protein? Let's get into the steps of how the information from your DNA actually gets created into a useful protein. So step 1, which is kind of step 0 because it happens before everything, is that every single cell needs to have a copy of this information. It needs to have a copy of the DNA. And so here I'm drawing a cell that's about to split into 2. So you're not going to see a nucleus, but you're going to see 2 sets of DNA because this is the part where DNA has to copy itself so that when it splits into 2 cells during a process called mitosis, each cell is going to have a copy of its own DNA. And the way that your body's cells do this is it takes advantage of the fact that DNA has 4 bases and each base has a partner. The 4 bases that you have are going to be represented by letters. The first one is the letter C, which stands for a base called cytosine, and its partner is the letter G, which stands for a base called guanine. And so whenever you see a C, it's going to pair with G, and vice versa. So whenever you see a G, it's going to pair with a C. The third base is called adenine, and we're going to use the letter A for that. And adenine's partner is the letter T. T stands for thymine. And so same thing with the other base pair. Every time you have an A, it pairs with a T. Every time you have a T, it's going to pair with an A. So these are the rules that your body follows when it is pairing up DNA. So let's say you have an original strand of DNA, A, G, T, C, and it needs to make a copy. It's just going to use the base pair. So the copy is going to have T, C, A, G. And now you've got 2 copies of this information. So in the next step, we have a cell and it's now ready to do its job. And so it needs to make some proteins. And so the first step it needs to do is the DNA needs to send its instructions outside of the nucleus. The DNA can't leave the nucleus, but all the stuff that makes proteins are located outside the nucleus. And so DNA is going to use a very similar structure called RNA. And that's this blue strand here. And so DNA is double helixed, which means it has 2 backbones, and RNA is different because it's a single helix. It only has 1 backbone. A lot of you have said it looks like half a ladder. And it also follows base pairing rules. So the one that's the same is that C and G when you're going from DNA to RNA are still going to pair with each other. But the difference is that when you have an A, it's going to pair with a different molecule called uracil, which is represented by the letter U. And then when you have the letter T, that still goes to the letter A. So really the only difference is the A to U partnership. So now we're going to practice transcribing DNA, which is the purple strand, into RNA, which is going to be drawn by a blue strand. So remember, this is happening inside the nucleus of a cell, and if we get a DNA sequence that is TAC-GTG, then the RNA sequence is going to copy this message by pairing with it, and it is going to end up with a RNA strand of AUG-CAC. And so now we have this strand of RNA. And the RNA now leaves the nucleus, and it's going to get read 3 bases at a time. This is important. And so the 3 bases, they are called a codon. So that's just a set. So this first 3, AUG, that's 1 codon. And the second set, CAC, the next 3, it's another codon. And so by reading the RNA 3 at a time, you can now translate them into amino acids. And so when you're translating, kind of like when you're using Google Translate to go from one language to another, we're going to be using the codon wheel as our translation tool. So let's deal with this first codon, AUG. If you look at the codon wheel, you find the letter A in the first circle, U in the second circle, and G in the third circle. It will lead you to the amino acid methionine, or MET for short. If you use the same rules for the second codon, you start with CAC, the C in the blue circle, A in the yellow circle, and C in the green circle. That's going to lead you to the amino acid histidine, which can also be written as HIS. And so now we've gone from the genetic code of RNA, and now we're starting to make a protein structure. And so your cells are going to continue to read codons and put a bunch of amino acids together, and maybe you start with methionine, and then you get histidine, then leucine, then serine, and proline. So these are all amino acids, and if you connect a string of amino acids together, you start getting the basic structure of a protein. And so that is how you go from the information from your DNA that transcribed to RNA. It made this string of amino acids until it eventually makes this protein, maybe this protein that heals your skin wounds. And so that is a summary of the central dogma of biology.